Welcome to Chapter 4. Having talked about the atoms and molecules in our previous chapters, we are now ready to move one step larger in our organization of biological structures. This module is going to be all about cells. And I know that quite a few of you were very interested on this topic, and it really is one of my favorites too. So we will have fun. The topic of cells and this chapter 2 is a very large concept, but it is also a very important topic. The technical development of scientific tools, however, was a key for making advances in the study of them. Dutch father and son optician team Hans and Zacharias Janssen are often credited as the first to put together something like a first compound microscope. It had a rather low magnification power, something like 3 to 9 times. In mid-1600s, Englishman Robert Hooke made many key observations of microscopic biology with what you see here as the early microscopes, which were really not much more than simple optical lenses. Then in 1670, Dutch merchant Anton van Leeuwenhoek upped the game a bit by making very careful first biological observations with a more advanced version of a microscope. This microscope was now able to have a magnification of 270 times. A huge jump from the early versions, isn't it? The fact that he was a merchant is indicative that the microscopes were very expensive at the time and really not accessible for the most. So he was one of those who was privileged to have the means to study microscopic biological matter. The fact that proper tools were not available led into all sorts of strange beliefs. Like for a long time, it was pretty commonly believed, for example, that microorganisms appeared spontaneously from lifeless matter. Of course, for us, this is clearly now as not being the case, but think about it, if you have no better tools for checking this, it is not an unreasonable assumption. And then in 18th century, more technical advances were made and microscopy became more common amongst the scientists. Perhaps some of the biggest improvements were made by English surgeon Joseph Laxon Lister in 1830s. His method of using multiple weaker lenses allowed getting around the problem of an image being blurred, as was often the case. And of course, today's microscopes are hugely advanced when compared to those. Really, using electrons instead of the light has been one of the key advances, as light has limits to the resolution of microscope. But we will talk about that a bit later, so let's compare light and electron microscopy. Light microscopes use beams of visible light to illuminate the object that we are examining and produce a magnified image. This is why the samples are often thin if we illuminate them from the opposite side. We can have a single lens light microscope where a single lens is used to magnify the object, or a compound light microscope that uses 2 lenses. As I said earlier, after the invention of a light microscope, a whole world of possibilities opened up. Significant advances were made in the science. If you look at the sample image that I have provided on the right, it is obvious that we can start to differentiate general shape differences between different types of cells, and we see nuclei quite nicely. On a typical H&E staining, they usually show up darker. The other image of a plant leaf on the left-hand side makes a good example. Those green blobs that you see are chloroplasts, a structure that you will become more familiar with later in this chapter. With a light microscopy, you can tell the shape of a plant cell and see that there are those chloroplasts. But that's about it. Electron microscopes instead allow us to go much smaller detail by using beams of electrons. Remember those guys that we spoke about on our chapter of atoms to illuminate the object. These are much more complex apparatus, and I do not expect you to learn in detail about how they work. But as a fun curiosity, you might want to know that whereas we use optical lenses to bend the beams of light in light microscopes, we now use magnetic fields to bend the beams of electrons. These electron microscopes can be classified at large in two groups: transmission electron microscopes, known as TEMs, and scanning electron microscopes, known as SEMs. The difference between the two is that TEM shows us a view through the sample, whereas SEM shows the surface of a sample. You can see some surface structure imaging examples of a variety of pollen on the left image. Oh, and one more point that I want to highlight. Electron microscopy images are really black and white. However, often you can see colored versions of those, but these colors are only added afterwards to highlight different structures. They are not real colors acquired with this imaging modality. Now for the fun, let's look at the difference of two pictures that we have seen before. The level of detail of a chloroplast shown with a light microscope on the left and electron microscope on the right are quite striking. So whereas the resolution of a light microscope is typically millimeters to the maximum of nanometers, electron microscopes can go much beyond this, all the way to the scale of 0.1 nanometers, which is enough to start to see more detailed structures within the cell organelles. Of course, the physical size and operation skills and costs between the two are hugely different. In our labs, we always have light microscopes, but it is only usually at the universities where I work where I have electron microscopes available. So here is a nice diagram that goes with what we said about the limits of what can be seen with different methods. With a naked eye, you can get to the scale of seeing some of the larger cells, but you would really need a light microscope to see more structures inside them, and definitely an electron microscope to see more detail about these cell organelles. I realize that unlike us Europeans, you guys probably were not brought up with a metric scale. And although it is not something that you might be necessarily familiar with, I am afraid that if you are going to do sciences, you will have to become familiar with it. I promise it really is quite simple and makes much more sense than the imperial scale. To move from one unit to another, you simply multiply or divide the values by a tenfold. I guess that the next thing that we notice is that cells are small. Remember, we needed all these special tools to see them. But although there are some exceptions, why cells typically tend to be so small? Well, here is the reason. Remember from your lab, materials must come in and leave the cell through its outermost layer, the cell membrane. You looked at simple passive diffusion as one of these methods. Well, there is a concept called surface area to volume ratio. What it basically tells us is that when the size of a cell grows, its volume increases much, much faster than its cell membrane surface area. Therefore, in proportion, it will have less chance for the material to get in or out. So it makes sense to keep the cell size small. Another way how you can look at this is that the transportation distances inside the cell would soon grow way too large to have materials reaching their destination before they are consumed by whatever is carrying these materials. Think of a fuel truck. If the distance that it has to cover to transport the gas is too long, it will end up having to chip into some of that fuel that it is transporting. And if the distance is way too long, it is going to be a bit of a pointless task, as all of the fuel will have been used before it even gets to the destination. So we will finish here for now, and our next section will move on to look at the cell membrane and its many adaptations in more detail.